Okay, so let's see. So this is odd. So up next we have Taiowa Montgomery from Colorado State University, and he's going to tell us about ALG-5, another of these uncharacterized Argonauts and C. elegans. Take it away.
Thanks to the organizers for this opportunity. You'll probably notice a lot of thematic and technical overlap between my talk and Julie's talk. Similar to Julie, we're interested in these Argonauts. To the right here in this figure is human AGO2 with the small RNA bound to it, and then on the bottom here also with an mRNA bound. I'm going to focus on the microRNAs. These are small RNAs that derive from these characteristic hairpins and guide Argonauts to unrelated mRNAs where they affect gene silencing. Now, as Julie mentioned, there's 3 subfamilies of Argonauts. The majority of microRNA-associated Argonauts are actually in this AGO subfamily. Clearly, Vis1 is an exception to that rule. This is the AGO subfamily in humans, flies, and worms. And so in C. elegans, there's 5 AGO subfamily Argonauts, 2 of which, ALG-1 and 2, bind microRNAs, 2, ALG-3 and 4, bind a subset of siRNAs, and there's a 5th member, ALG-5, that hasn't been characterized. But its relatedness to ALG-1 and 2 suggests that it might bind microRNAs, or perhaps similar to ALG-3 and 4, it binds siRNAs. And so this is, of course, fairly easy to test. And as Julie demonstrated, we can simply pull down these protein small RNA complexes, isolate away the small RNAs from the Argonauts, and sequence them. Okay, so we did this with ALG-5 and in parallel with ALG-1 and 2 because of their relatedness to ALG-5. For the sake of time, I'm going to focus on our ALG-5 data. So which small RNAs are bound to ALG-5? So in this Western blot image, on the left-hand side, you have the ALG-5 protein in our crude cell lysate that we use for IPs, and on the right is the protein in our immunoprecipitate. So we isolate away the small RNAs from these 2 fractions, and we subject them to sequencing. Now, to account for contamination of small RNAs from the cell lysate in the IP fraction, we look for enrichment of small RNAs in the IP relative to the cell lysate. And what we find is that microRNAs are enriched around 2-fold, but every other class of small RNAs is depleted. So this places ALG-5 in the microRNA pathway. So as a class, microRNAs are enriched in ALG-5, but let's look a little more closely. So in the scatter plot, we're plotting small RNAs as a function of the number of sequence reads in the IP relative to the cell lysate. And you can hardly see the shading here, but small RNAs that are in that shaded region off to the right, of course, would be depleted in the IP, and small RNAs that are enriched in the IP will be on the left-hand side of the plot. And interestingly, the majority of microRNAs shown by these red dots are actually depleted in ALG-5. There's only really a small subset of the roughly 300 microRNAs in C. elegans that are actually enriched in the ALG-5 IP. But some of these are very strongly enriched, several of them greater than 50-fold, and we don't typically see this with the other microRNA-associated Argonauts, ALG-1 and ALG-2. And of the 25 or so microRNAs that are enriched in the ALG-5 IP, 6 of those are uniquely bound to ALG-5, and the remainder are also bound by either ALG-1 or ALG-2. And we see considerable overlap between the microRNAs that are bound between each of the 3 Argonauts. Now, Argonauts affect gene expression. What is the role of ALG-5 in regulating gene expression? To test this, we did RNA-seq of wild type and ALG-5 mutant animals. And we analyzed the data using 2 common differential gene expression analysis methods, CUF-DIF and DE-seq. And the methods were largely in agreement with one another. However, by cuff-diff, we identified substantially more genes that were upregulated as well as downregulated in ALG-5. Now, within the upregulated gene set, male function genes, particularly sperm function genes such as SSP10, shown here to the right in this little genome plot of SSP10, were significantly enriched regardless of whether we used cuff-diff or DE-seq, or if we simply looked at the overlap between the genes that came out of those two methods. My shading got a little bit washed out there. That should be a pie chart with a big gray slice that you can't see. And within the downregulated gene set, cell structural and muscle genes were significantly enriched. Okay, so ALG-5 binds a subset of microRNAs. It affects a handful of genes. Where is it actually functioning? So the fact that we see upregulation of male function genes suggests that maybe it's functioning in sperm. And consistent with this, we see ALG-5 protein expression peak in late larval stages into adulthood. And as you can see in this little cartoon at the bottom here, these are the stages where the C. elegans germline, shown in gray, starts to rapidly proliferate, and of course sperm and oocytes form. And consistent with ALG-5 being expressed in the germline, in GLP-4 germlineless animals, we see about a 400-fold reduction in ALG-5 mRNA levels relative to animals that contain a germline. And this is actually the first example I'm aware of a microRNA-associated Argonaut being specifically expressed in the germline. Now, surprisingly, though, if you look back at that blot panel, ALG-5 protein is actually depleted in males, which of course contain sperm, relative to hermaphrodites, which of course contain both sperm and oocytes. And if we look at ALG-5 expression across germline development, what we find, and this is based on the Ranke Lab's microarray profiling data, what we find there with the ALG-5 in orange is that its expression peaks during oogenesis, and it tracks with the oocyte-specific siRNA Argonaut ergo-1, and inversely with that of the sperm-specific siRNA Argonauts ALG-3 and 4. So this suggests that ALG-5 actually functions during oogenesis to repress male function genes. So you're probably wondering what the phenotype of ALG-5 is, and that's something that we're actually still exploring. But what we've observed so far is that it has a modest reduction in fertility, just based on the number of viable progeny that it produces. Similar, so on the right-hand plot, you can see that, and it's similar to what we see in ALG-2 shown there on the left. Not nearly as dramatic as what we see with ALG-1, but it's actually somewhat additive with ALG-1 as well as with ALG-2, which I'm not showing here. So in ALG-1 single mutants, we see about 50% of the animals being sterile. In an ALG-5, ALG-1 double mutant, we see greater than 80% of the animals being sterile. So ALG-5 binds a subset of microRNAs. It's expressed in the germline during oogenesis. It represses male function genes, and it's required for optimal fertility. And so we think this points to a likely role for ALG-5 in helping to specify female fate to germ cells during oogenesis. Now, I didn't have time to talk about our ALG-1 and ALG-2 results, but I'll just sort of briefly summarize some of our findings. So ALG-1 binds most microRNAs with similar affinity. We don't see the types of bias we see with ALG-5, and it's highly expressed throughout development, consistent with it having a very central role in the microRNA pathway. ALG-2 also binds the majority of microRNAs, but it has a particularly high affinity for the miR35 family. And the miR35 family is interesting because it's one of the few families of microRNAs that's absolutely essential for embryogenesis. And ALG-2 is predominantly expressed in embryos. And so we think ALG-2 is providing robustness to the microRNA pathway during embryogenesis. Its function is actually somewhat redundant with ALG-1 during embryogenesis, as well as into larval stages of development. And so this is the lab. I talked about Kristen's project today. Just a quick advertisement. We're looking for postdocs and graduate students, so if you're interested in living in beautiful Fort Collins, Colorado, please visit our lab website, montgomerylab.org, or check us out on Facebook. Thanks.
Okay, we have time for plenty of questions. Ty has 4 minutes left. He gets the award for being on time.
People always appreciate it if you're just a couple minutes short on your talk.
Yeah, good job.
Do these microRNAs that are specifically bound by ALG-5 have some sort of shared structure that might explain how ALG-5 specifically binds to them?
That's funny because I had almost the same question for Julie, but for the sake of time, I wasn't able to ask it.
We can talk about it later.
For sure. And the answer is not that we've been able to identify. My guess is that it's more of a spatial and temporal thing where ALG-5 is specifically expressed in this very limited set of cells in which there's a very limited set of microRNAs which it can bind to.
Thomas.
Yep, I'll indulge since there's a bit of time. So do you know if it's expressed in the Z1, Z2 precursors in the embryo?
That's a good question. We have an HA fusion of ALG-5, and in the past we haven't had very much success immunostaining for HA. So we're actually making a GFP-based transgene so that we can actually address that sort of question.
All right, I just wanted to check also, make sure in your conclusion you had references to affinity. And I'm playing with the 35, 45 quite a bit. Do you mean biochemical affinity or just mean that it's enriched in the IP?
Yeah, that's a good question. So if you look at, like, let's say ALG-1, pretty much all microRNAs bind to ALG-1, all the guide strand microRNAs. And they're rarely ever enriched more than like threefold. And that's about as much as you can get enriched if you bind everything, because about a third of the small RNA population is microRNA, so that you're sort of capped at a threefold enrichment. But with ALG-2, it binds, the majority of microRNAs are enriched in the IP, in that same range, 2 to 3-fold. However, the miR35 family is all enriched greater than 10-fold. There's a couple other embryonic families that are also enriched.
Okay, so your argument is about enrichment.
Yeah, it's a good point. Yeah, it's not really a biochemical affinity.
I was curious because the pocket might be different. You might actually have sequence preferences.
Yeah, absolutely.
All right, okay. Yeah. Thanks for clarification.
But I suspect it is also related to like ALG-5. It's also more of a temporal, spatial thing.
Yeah, I agree. Yeah.
I wonder if the ALG-5 and to a larger extent ALG-1 and 2 function in sperm or oocytes, in reducing the brood size.
Very good question, and we are in the process of doing those experiments to specifically test that. Our hypothesis is that it's functioning in oocytes, but we're actually doing experiments right now to specifically test that.
Ty, I have one question for you. Did you see any 26G RNAs coming down with ALG-5? So it's in this group that is, closely related to ALG-3 and 4, and they bind to 26G RNAs. So you didn't see any, right?
Right, yeah,
Okay.
we did not see 26G RNAs enriched in the IP. I will say that we did actually see a very small number of piRNAs that appeared to be enriched, but given that it seems to be expressed in the germline where there's a lot of piRNAs, we were sort of concerned that might just be sort of a contamination issue where the piRNAs are just sticking to the protein bead complex.
All right, great. if there aren't any other questions, we'll move on to the next speaker. Thanks, Ty.